Hello, good morning, how are you? It's a pleasure for me to stay here in this webinar about public mental health and neuroscience. Today I will talk about my main project regarding the G9a methyltransferase from transgenerational epigenetic inheritance to the discovery of new G9a inhibitors for Alzheimer's disease treatment. As it is well known, Alzheimer's disease is the most common cause of dementia, representing about the 16 to 80% of the total cases. Is a progressive and irreversible neurological disorder characterized by cognitive decline, memory loss, beta amyloid as a AD neuropathological hallmark, and phosphorylation, the tau phosphorylation. Furthermore, the main risk factor is the aging, in which all of these events, DNA damage, cell membrane attrition, loss of proteostasis, among others, are really important. Among them, epigenetic alteration as one of the primary causes of this cognitive. For this reason, it's important to know what is epigenetic. Epigenetic was described for the first time by Conrad Whittington in 1914, and he describes the epigenetic as the studies how your behavior environment can cause the change how your genes are expressed without any change in the DNA sequence. To do this stuff, the epigenetics use mainly three epigenetic mechanisms that are DNA methylation, histone modification, and non-coding RNAs. Regarding the histone modifications, there are methylation and acetylation. With these two marks, the 3D structure of the chromatid can compact or relax. So, furthermore, it is well described that epigenetic alterations can promote changes in learning and memory formation. Due to this, one of them, the G9a GLP complex, is an important complex that can regulate the memory formation through the repressive mark, the H3K9 methylation 2 and 3. For this reason, epigenetics in the last 2 decades, it was really important topic in which a lot of papers appeared, mainly in 2010. So, epigenetic is the connection between these three important factors: the aging, the environment, and the genetics. So finally, as is well known, Alzheimer's disease is a multifactorial disease. It's a really complex disease. So due to this background, in the last decade, I performed my PhD about how epigenetics can modulate the cognitive decline that appear in two AD mice models, the SAMP8 mice model and the 5xFAT mice model. The two well described in the bibliography as a AD mice model, and I found important changes in the main repressive marks from epigenetics in both models, and changes in the main methyltransferases and translocases enzymes, the machinery who can regulate these marks, these epigenetic marks. To this, I found several targets, and one of them was the G9a methyltransferase, a lysine methyltransferase that can add this repressive mark in the histone H3K9, can add methylation, methylcytosine molecules, one or two each, and then can inhibit the the expression of neuronal genes and promoting the AD hallmarks. A few years ago, start to appear different evidence regarding this G9a GLP complex, in which the inhibition of this complex promote amelioration in the main AD hallmarks, such as beta amyloid, and promoting at the same time synaptic plasticity, but always in the in vitro studies. Due to this, in 2019, I performed my first in vivo study in 5-fixed model, in which I demonstrate that the pharmacological inhibition of G9a restore cognition and promote changes in the main neuropathological events such as oxidative stress, neuroinflammation, reducing beta amyloid, and promoting amelioration in this in the cognition in this 5-fixed model. So this article, it was so important, published in the Aging Journal 2 years ago, and this our proof of concept that the inhibition of this G9a could be a promising target for Alzheimer's disease therapy. For the other part, it is important to know that the main cause of Alzheimer's disease is as I told you, is the aging, and it's not well explained then the inheritance of this multifactorial disease. It is clear that it's an sporadic and multifactorial, and only a little cases can be explained as a familiar inheritance regarding a specific mutations in the APP presenilin, so basically in the amyloid processing pathways. So here it is the behind this there is an epigenetic deregulation as well. So we can explain if there is a inheritance, epigenetic inheritance is more important than genetic inheritance. So to demonstrate this, so to explain a little bit more about this, we recently published a review in which we explain the contribution of epigenetic inheritance process on age-related cognitive decline and Alzheimer's disease, and here we demonstrate several evidence that we can explain why we can think about this epigenetic inheritance take part in the age-related cognitive decline and Alzheimer's disease. Something that is not really well described, at least in human and only in C. elegans model and mice. Basically, there are a few evidence. So one thing more that we can demonstrate, and one of our project is if it's does Z25, the ortholog of G9a in C. elegans play an important role in epigenetic inheritance in cognitive impairment after environmental harmful issues. So to perform this kind of experiments, we use the C. elegans is a little worm that I explain after. So why we use C. elegans? Because it is important to know that our brain is too much complex. So there are billions of neurons. Each neuron present more than 30,000 synapses. So it is good to work with our really complex brain. So what is important to know for why to use this C. elegans, because it's a powerful model for studying memory, because it's much simpler brain, only 302 neurons, just 7,000 synapses, and it is well known the connectome, short life, and many molecular pathways are similar than humans. So things that that confirm that this C. elegans it's well to use for this kind of experiments, mostly for neuroscience. So to perform this experiment regarding the transgenerational epigenetic inheritance, we perform an imprinting with this model. But what is imprinting? Imprinting basically is long-term memory that appear in a specific time, in a critical period, and is a adaptive behavior that responds basically for changes in the environment, sensory cues, and so on. So basically is after the birth in the L1 to L4 period in the C. elegans. This imprinting allow the adult behavior patterns. So what they perform here? Here, the experimental paradigm design is basically to promote early life toxic stress after anti-missing treatment. This experimental paradigm is a collaboration with Chava Group from University of Semmelweis. This experimental paradigm is basically to perform early life toxic stress with antimycin in the Escherichia coli, and after that they remember the C. elegans form. When they are adults, they remember that the Escherichia coli is a toxic feed. Basically is this. So here we use two kind of strains. One of them, the N2, to know what happens with the behavior, and the other is the HSP6 GFP transgenic C. elegans strain, to know if the body remember this imprinting. We use these two strains because the HSP6 GFP strain it doesn't work correctly or properly for the chemotaxis assay. So basically, our results confirm the behavioral molecular inheritance after early life toxic stress until the F4 generation. So the F1, the F2, and the F3 remember that Escherichia coli was with antimycin, and they prefer to avoid the Escherichia coli and go to the Bacillus subtilis. In the chemotaxis assay, we obtain the same results. And finally, this something new and something interesting, something really relevant and really cool, is that this after this imprinting with after this early life toxic stress, we obtain an overexpression of this Z25 gene that along the generation will disappear. So basically there is a correlation between the behavior, the HSP6 expression, and the Z25, demonstrating that the Z25 modulate this molecularly this. Furthermore, we obtain really interesting results, preliminary results. We use two strains, two knockout strains with Z25 and MET2, and we obtain relevant results regarding the Z25. We demonstrate that without Z25 expression, this imprinting disappear, something that doesn't happen in the another important insight about the Alzheimer's disease is the drug development. As it is well known, recently appear a new treatment for Alzheimer's disease, but is a little bit there is a little bit controversy if finally this aducanumab finally works, but doesn't matter because it's approved by the FDA. But finally, currently there is no cure for Alzheimer's disease. So another important insight is that during these years, the drug development for epigenetics it is more and more important during the pass of the years. Right now, there are more than 5 clinical trials for epigenetic therapies. So here appear again our purpose is if G9a GLP inhibition could be a potent therapy for Alzheimer's disease. To perform this experiment, we use the SAMP8 mouse model generated by phenotypic selection in which there are more than 11 strains. But finally, what is important here is that SAMP8 is suffered fast senescence, has learning and memory deficit, present mainly AD hallmarks, as amyloid processing alteration and tau phosphorylation, increased oxidative stress, and loss of synapse and neurons. So it is a really good model to perform intervention for drug discovery, in which you can test it your small molecules, for example, and so on. And in addition, recently during these years, I published several articles in which I demonstrate that epigenetics can explain in part the senescence phenotype that characterize this SAMP8. So another important insight that is really interesting to know if you want to test novel epigenetic drugs for Alzheimer's disease. So, as I told you, as aforementioned, we performed this proof of concept for with this UNC0642, well-established G9a inhibitor with a really relevant IC50, less than 2.5 nanomolar, not too much BBB penetrant, but finally it works. So we decided to perform this experiment, and it was good. So after this experiment, in which confirmed this well proof of concept for Alzheimer's disease therapy, we want to go deep in what molecular changes promote this G9a inhibition, and to do that we use now the SAMP8, and we perform different next generation sequencing experiments like RNA-seq and G9a-seq, basically, to find different molecular pathways that can be modulated by this G9a inhibition. Importantly, to confirm that this treatment was well done, we evaluate the cognition, and again we found amelioration through novel object recognition in short and long-term memory, as well as we found a reduction in this repressive mark, epigenetic mark, the H3K9 demethylated, confirming that the pharmacological intervention was, as I told you, we performed the RNA-seq and G9a-seq. We found relevant pathways that were enriched related to regulation of histone modifications and AD neuropathological hallmarks, as well as important different expression in different microRNAs. Promoting a transcriptional profile with beneficial effects on cognitive performance. So it was a correlation because with our results in behavioral and molecular studies. So one important change was the GMFB GMFB pathway enhance the neuroplasticity status, and is a new pathway involving neuroplasticity associated with this G9a inhibition. So it is again a new important demonstration that why it can be useful this G9a inhibition. So GMFB promotes changes in phosphorylation in PIT38, increasing the CREB, and finally increasing neurotrophins such as BDNF, for example. So here in the right you have the Western blotting bands, and are really clear. As you can see, the GMFB, PIT38, phosphorylation, phosphocreb, and so on. So it are really. So for the last experiment the last project that we have in our hands is the drug discovery process for new potent G9a inhibitors. So there is a really problem in these G9a inhibitors. For example, the VX1209 is really toxic and only works in cells. And for example, the UNC0638, it doesn't cross properly the ED, and the UNC642, the same thing, and just only has a better pharmacokinetics. So it is important to obtain new potent G9a inhibitors that we can serve for Alzheimer's disease treatment. Due to this, we perform a virtual screening. We obtain more than 10 new compounds, which we perform the AC50, and for we obtain subnanomolar AC50. So this is the first time that is described new potent G9a inhibitors with this AC50, subnanomolar AC50. After this, we perform with this C. elegans transgenic strain, the CL2006, with that overexpress amyloid beta in muscle cells, and for this reason they suffer paralysis. So it is a really good model to perform concentration response, and then we use this to confirm if our novel potent G9a inhibitors are better than the well-described UNC0638, and we obtain really good results for all of them. In order to confirm that can be used for Alzheimer's disease treatment, and for link these results with the AD pathology, we perform the theophylline staining in order to demonstrate the reduction of beta amyloid aggregations. Here you can see the reduction in the well-established control, the UNC, and we have two compounds that are better than this well-established control in more than 50%. So we have a really good results, amazing results. So with this new offers the opportunity to design and synthesize new structures that maybe can be. So basically here we did a structural-based virtual screening. We performed the in silico CNS drugability analysis. We performed the PAMP assay and in vitro G9a inhibition to show the EC50, and finally we performed the in vivo with C. elegans. And as I told you, really potent with nanomolar IC50, really good BBB permeability, and reducing the deposition of beta amyloid in more than 45%. So really cool. And as a conclusion, it is important to know that we demonstrate the proof of concept that inhibition with this UNC0642 prevents beta amyloid accumulation, increases synaptic plasticity and neural markers that are characteristic of AD. Moreover, this compound was able to reduce oxidative inflammation. This was demonstrated in 5 years lab. Here, as a new approach with SAMP8, we demonstrate after the inhibition of G9a, the activation of the GM pathway and neural plasticity status on SAMP8, and we design an optimized new candidate as inhibitor of G9a with novel structures providing both leads and G9a inhibitor designed and demonstrating the participation in AD pathology. And finally, that early life toxicity and stress, so the imprinting experimental paradigm, can offer us the opportunity to demonstrate that SET25 can participate in this transgenerational epigenetic inheritance, promoting changes in molecular and behavioral status in this C. elegans that finally promote cryoprotection. I want to say our message is that our work reports a new finding that pharmacological inhibition of G9a may be a promising target for Alzheimer's disease therapy, promoting neuroprotection through reduction of its repressive chromatin marks, as well as it takes part in the epigenetic inheritance memory process. Finally, I want to thank to my mentor, Dr. Anessa Calvas, who provide me all that I need during these years, and we share our love for the science and motivation, as well as Aina, that is doing her PhD regarding the participation or mention in the epigenetic inheritance process and Alzheimer's disease, and the rest of my group, Julia, Pine, Shan, Fotini, as well as Vanessa. So thank you so much. I hope you love this presentation, and thank you so much. Bye bye.